Yeah, today our topic is the laboratory consumables, especially when we are working in a microbiology laboratory, which things we require for our testing, which items, which consumables we require for our testing laboratory. This one is the summarized view in which we needed different items for our laboratory functions. Hopefully, it will be grateful for you when you will watch all this video. The next slide, this one is the laboratory equipment in which this slide we already discussed these topics in our previous video. I will share the link of this video in our description sections. If you want to visit this one, you can go to the descriptions and visit this video. Now we will discuss our consumables. What are the consumables that we record in our laboratory one by one? First one is the PPEs. PPE is personal protective equipment. This one is the most important need for any laboratory in which we needed glove, face mask, head covers, and mask, face mask. Just like all these things we needed for our shoe cover, maybe designing the person. This one is for personal safety and this distilled water because ISO methods claim that our distilled water have 5 microsimol per centimeter square conductivity and less than 1,000 maximum allowable limit is less than 1,000 CFU per ml for distilled water. And others is the glasswares that includes different glasswares. We needed beaker, we needed cylinders, we also needed screw cap bottles, plastic petri dishes. The most important is just to we need to use disposable petri dishes. Then we use cotton bundles, tissue roll for our cleaning purposes. For ethanol is also using as in disinfectants. We need to clean our workbenches. We need to clean our cabinet. We need to clean our even instruments for by using the ethanol 70%. Spatula is normally used for weighing purposes for samples of water media. Test tube is very important because when we go for any broth preparations, we need this test tubes for our even for confirmations for our even to any gas productions, we need the test tubes with the Durham tube. We will discuss in the next. Scissors, this one is used for sample cutting. We will use sterilized scissors and autoclaves and then you use it for our sample processing. Culture loops for bacteria transfers and inoculating needle. This one is normally used for stabbing of and culture media, just like we want to check our motility. We need to, for the glucose fermentation, we will simply take calumnies, we will stab in the respective agar. Burner is required. We'll use any platinum base loop. We need burners for sterilization of that loop and then we use that loops. Burner is also requirements. Thermal tubes. This is normally used for checking the gas productions in any broth mediums, especially in the form of bubbles. Swab 6, this one is also required for the environmental monitoring. When we go for checking any incubators, any machines, any equipments, we will simply take a swab and then we will process and our testing. Separator. You know, as we already know that ISO methods are based upon two techniques, pour blade and separate plates. But when we go for the separate plates, separate is requirements. We will use glass waste, we will use also disposable. Autoclave tape, this one is used for autoclave verification. Either autoclave cycle is completed perfectly or not. Biohazard bags, this one is very important thing, especially when we'll decontaminate our media, our items, our plates, used plates, we will use these biohazard bags. Aluminum foils for any kinds of sterilization, we will grab our materials in the aluminum. So mature bags. When we want to go for any samples to process, we simply take our sample in these pre-sterilized bags and place in their holders. Tray is normally used for shifting of materials from one place to another. Micro tips, as we already discussed in our equipment section. Micro pipettes, we needed the tips required for that. This one is the pipette fillers. This one electronic pipette. We will check and release according to our requirements and different volume-based papers we will add. Glass papers normally used in this filler. Glass slides and grams are normally used for the gram setting in which as for also in microscope is required for checking bacteria is gram positive or gram negative. Up and off, this one is normally used for culturing for storing of our positive cultures. We will make glycerol stores in this bundle of tubes paste with. As we already discussed, this we needed biohazard bags. We will autoclave and then we use that biohazard bag in this biohazard waste bins. Parafilm, this is normally used for sealing, just like in the bundle. Will prepare positive cultures for sealing purposes. We use the parafilm. Thank you so much for watching. If you have any questions, any suggestions, you can add in the comment section.